Hey, how's it going, guys? I hope this video finds you well, as always. In today's video, we are going to talk about the central dogma, okay? And that is protein synthesis. So just to get you started, we're going to look at a warm-up question. The warm-up question says, the sequence of nitrogenous bases varies widely. The sequence of the bases in DNA is most important for which of the following? It says, providing the instructions for the trait of an organism. Is it preventing mutations from occurring during DNA translation? Allowing the DNA to have the shape necessary for replication? Or helping form the sugar phosphate backbone of DNA molecules? So make sure that you answer the question once that video stops. And the best answer for this question would be choice A. Okay, the most important thing about DNA are the sequence of those nitrogenous bases, and those are important because they provide the instructions for the traits of an organism. So A is the best answer in here. Now we're going to look at the objective. The objective is to explain the purpose and process of transcription and translation during using models of DNA and RNA. Now, basically what we need to be able to do is be able to understand the difference between transcription, what happens there, and then translation, also know what happens there. So make this into an essential question. The easiest way is putting that can I statement at the very top. So can I explain the purpose and process of transcription and translation using models of DNA and RNA? And then you can put that question mark at the end. Okay. All right. Some I can or I know statements that you need to be aware of for this specific lecture, just things to keep in mind. Know the central dogma of biology. We're going to talk about what central dogma means in this video. It's very simple. I can explain the process of transcription. Again, this is one of the things you need to be able to do. I can explain the process of translation. So those three are important to know. And then you have some things that are from different topics that we have covered. Like, for example, this one was covered when we did biomolecules. I know that amino acids are the monomer of proteins. Well, remember that amino acids are what make up the proteins. And I know that ribosomes are organelles and they synthesize proteins. You need to remember what that word means, and that word means to make or to build. Okay, please remember what that word means every time that we use synthesize or synthesis. All right, so what is the central dogma? Basically, the central dogma of biology states the process of making proteins, how we go from DNA, transcription makes RNA, and then translation makes protein. Now, where does these things happen? Here, this is going to happen in a different place than this process. So we'll go over that in the video. But central dogma basically means the process of making proteins. DNA and RNA, what's the difference? Well, this is just a quick review. Remember that DNA and RNA both have nucleic acids and their monomers are nucleotides. Okay, now the difference is that RNA is single-stranded and it has a nitrogenous base called uracil instead of thymine. So make sure that you write those quick things down, but we have covered this already. Now there's, well, two main types in this PowerPoint, but you need to remember the other type that we talked about in class, where there's messenger RNA, which carries instructions from DNA to make proteins. There's also transfer RNA or tRNA, which transfers amino acids to ribosomes during protein synthesis. And then don't forget the third one, which is ribosomal RNA, which is the one that we call rRNA, which is basically what makes up the ribosome. So ribosomes are made of rRNA. Okay. So remember proteins. Proteins are polymers, and these polymers are going to be made of chains of amino acids. Okay, the amino acids are the monomer for proteins. So they're going to be made inside of the ribosome. Now, these right here, each little circle will represent one amino acid, and they're going to be tied together by what we call peptide bonds. So each of these are going to be tied together. If you notice, there's a little line. Let's look at this one right here. There's a little line between those two. That's what we call a peptide bond. Okay, basically a protein bond that is going to tie all of those amino acids together. All right, and there's actually 20 different amino acids. All right, 20 different of one of them. This is just a quick question. Where can you find ribosomes? Can you find them in eukaryotes? Can you find them in prokaryotes? Can you find them in viruses? Can you find them in all of them? Make sure that you answer that question when the video stops. The answer is eukaryotes and prokaryotes. Viruses, they do not have ribosomes. Now, all living cells will have ribosomes so that they can make proteins, and these two are obviously living cells. Now, the two steps in making proteins. One is going to be called transcription. This is where we use DNA to make mRNA. And then the second step is translation, where we get that mRNA and we translate it into proteins or chains of amino acids, right? Where is transcription going to happen? Well, transcription involves DNA and it's going to happen inside of the nucleus. All right, this is where transcription is going to occur, inside of the nucleus of the cells. Because remember, DNA cannot exit the nucleus. However, RNA will take a copy of it and be able to exit. Let's take some notes about that. This is the first part. Transcription is making RNA from DNA. It takes place in the nucleus. These are the following steps. The step 1, DNA strand separates. So remember, it's going to separate into two separate strands like this. This is one strand here. This is the other strand there. And then RNA nucleotides bind to the separated DNA template strand, forming an mRNA strand. So basically, it's going to copy that DNA template strand, and thus you're going to have a mRNA molecule right here, which I'm pointing to using my mouse. Okay, so that's how transcription happens. Very simple. Not gonna worry about all the enzymes that are involved in all the things, but obviously you have to remember that there's a lot of different enzymes involved in breaking it apart and then putting all of these nucleotides together where they need to be. Okay, so this is just what it kind of looks like. Remember, keep in mind we're copying the template strand. So if you have a C on the DNA, and again, this is the DNA here, CAAT, well, that's going to be copied to GUUA, and so, thus far, all the way down the line. And then that is going to be called an mRNA, which is able to get out of the nucleus of the cell. DNA is going to stay in the nucleus. This is an animation that I'm going to put in Schoology. Okay, make sure that you click on it. I'll give you a little bit of time for you to make sure that you click on that and play around with it a little bit. It explains this process really well, so please spend a little bit of time to do that. I'm also going to have one for translation, so please make sure that you do that and look at them. So after transcription, the mRNA leaves the nucleus, like I said, and travels to the ribosomes in the cytoplasm. So the ribosomes are the only place to build proteins. Keep in mind that once it exits the nucleus, that mRNA will go to the ribosomes. The next step is the process of translation. And in translation, we're going to have different things happening. Remember, this is where we go from mRNA to proteins. And the way that this is going to happen is very simple. Well, it's going to take place at the ribosomes, which is this organelle that we see right here. This is just a diagram, but it's going to look like this. And then the mRNA is going to attach to the ribosome. So mRNA attaches to the ribosome like this. This is your mRNA. It attaches to the ribosome. And then tRNA carries the correct amino acid to the ribosome. Now this is going to be where tRNA comes in place. So this is tRNA coming in And one thing that is important to know is that tRNA is going to have an amino acid attached to it at the very top, like this. And so it's going to come in and find the complementary strand. So it's going to be like, if this is CUA, well, it's going to find whatever complements that, and that's GAU, and that's going to attach a specific amino acid to it. So once that amino acid is attached to it, it goes to the next step, which is CCC. Well, what attaches to CCC is going to be GGG. That's going to be come in here, and then those two are going to attach together, and you're going to form this long chain of amino acids, which is the actual protein. All right. So looking at a diagram here, very simple diagram, understanding the difference, this right here, would that be DNA or RNA? We'll look at what bases are present. You see those T's? That tells you that that is DNA. So when we go from DNA to something that looks like this, notice those U's there, okay? That's no longer DNA, that's something else. It's going to be mRNA. Well, from DNA to mRNA, we call that process transcription. Then from mRNA, which we just have it here, that mRNA is going to copy into or translate into amino acids. Well, that process, we call it translation. One more thing that I do need to point out is that this molecule that we see right here is actually the tRNA. Okay? So this entire molecule is the tRNA. However, those things that are attached to them are the amino acids. Okay? So those are the amino acids. The entire thing is called a tRNA molecule. All right? A little bit of more central dogma illustration here. Again, going from DNA to RNA, what do we call the process? Transcription. From RNA to protein, what do we call it? Translation. Okay? Now let's try to fill this out real quick before we go. Make sure that you fill this out in your notes as well before we finish up this video. So we have this molecule right here. What is this? Well, that is DNA. Then that same molecule of DNA is going to go through a process to make a copy of something that looks like this. It's single-stranded. So this single-stranded molecule is RNA. And where is this going to happen? Well, all of this is going to happen inside of the nucleus. Now, this process, we call it transcription. All right. Once transcription is done, you have this mRNA, specifically mRNA, is going to go out of the nucleus to the ribosome. So that's a ribosome there. And the next thing is translation, which is going to happen. All right. The entire process of making proteins is what you call translation. Now, this molecule still is mRNA that will attach to the ribosome. And once it attaches to the ribosome, it goes 3 units at a time, 3 nucleotides at a time. It reads them codon, 1 codon at a time, 3 by 3. And then it's going to leave one specific nucleotide, one specific amino acid for each of those codons that it copies. So those are amino acids that are being growing in here. And then the last thing is that this is happening in the ribosome. So once you have that chain of amino acids, then it can fold up into a protein. Okay? So that's going to conclude this video, guys. We're going to talk a little bit more about this topic as a class. As always, I hope you find this video interesting, and I'll see you on the next one. All right?